Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell. I'm going to be your host here tonight. I'm with my co-host, which is Marco Sonobi. Welcome, Dr. Sonobi.
Hi, Scott.
Yeah, appreciate having you here.
Thank
Our color commentator today is going to be Dr. Pete Hansen from the University of Florida. And our special guest is going to be Dr. Masru Sagheer. We're going to be discussing a presentation, Masru, that you're going to be giving here at the Florida Ruminant Nutrition Symposium. And I guess that'll be tomorrow,
This evening.
This evening.
5 p.m.
And that's going to be titled Feeding Rumen-Protected Choline During the Peri-Conceptional Period Programs Postnatal Phenotype of Calves. What I'd like you to do, let's do a couple things. First, just tell us a little bit about yourself. And then what I'd like you to do is kind of give us just kind of an overview of what your presentation is going to be like tonight.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
So I originally came from Pakistan, where I did my bachelor's in veterinary medicine, and I did my master's in animal reproduction, where I studied IVF in our native buffaloes. And I moved to US in August 2021 when I started my PhD with Dr. Hansen. For my PhD, we are working to see how can maternally derived molecules, essentially nutrients, can affect the embryonic development, and when you transfer those embryos, if or how they can affect the postnatal phenotype of the calves. And for this experiment that I'm presenting today at the conference or symposia, we have fed rumen-protected choline during the peri-conceptional period to beef cows, and we have looked at the phenotype of the calves, if that feeding can have an impact 9 months later at the calves.
Okay, very interesting. So exactly how long are you feeding it?
So we have some in vitro data where we supplement choline chloride in the embryo culture medium only for the first 7 days of embryonic development.
Got it.
Which is when the embryo is a one-cell zygote to the blastocyst stage of development. That is only 7 days in the culture dish. And we see that when we supplement choline during those 7 days of development and we transfer those embryos to the cows, we see that the calves, they are heavier at birth and at weaning.
Okay.
So we repeated that experiment 3 times now, and we have seen the effects on the weaning weight consistently. So the idea for this experiment stems from those in vitro studies, where supplementing choline have positive impact on the calves. So for the feeding study, we are trying to repeat the same in vitro experiment, where we are feeding rumen-protected choline 1 day before artificial insemination until 7 days after AI.
Okay.
So which covers the time period of ovulation, fertilization, and first 7 days of embryonic development.
Okay. Now, before we start talking about some of the results that you've seen, let's kind of go back to the in vitro stuff. So you were measuring weaning weight. How much larger were those calves?
So for our first experiment, we saw about 19, if I'm not wrong, about 17 to 19 kilograms increase in the weaning weights from the choline-supplemented embryos that became calves.
Okay.
Yeah.
And then did you follow those?
Yes, for our very recent study, one of my colleagues, Dr. Lainey Heyman, she took those calves to slaughter. and she
Okay.
looked at the carcass weight, and the effects were even seen on the carcass weight, and the meat was even better for
Okay.
those animals. So,
Okay. And have you done this on lactating animals or dairy animals?
so this was all beef cows,
beef
where
cattle.
we had oocytes coming from Brahman animals and Brangus animals, and those were transferred into mixed breed of cows, beef
Okay.
cows, all beef so
Right.
far.
Interesting. Dr. Hansen.
Yeah, no, Monster did a fantastic job. Yeah, we've done 3 experiments where we transferred embryos treated with choline or without choline. And the first experiment we did, we saw heavier birth weights. And actually, gestation length was a little bit longer in the calves derived from choline-treated embryos.
That causing issues with the heavier calves?
Well, the cattlemen were not very happy when
Yeah.
they saw that. They were not that much larger,
Got
but they're looking for smaller calves. In the other 2 experiments that we did, there was no effect on birth weight, no effect on gestation length, but we still saw this, like Mauser said, about a 17 to 20 kilogram increase in weaning weight.
Yeah.
Not statistically significant, but the effects are usually greater in the male calves than the female calves, and that's a characteristic of fetal programming, is that there's sex effects, that the effects of maternal environment on the male embryo are often different than on the female embryo. And we're not seeing a significant effect of sex on the response to choline, but it's kind of consistent in our studies.
Can you talk a little bit about mode of action of how this is happening?
So there are so many things that can happen when you talk about early embryonic development, right? Those zygotes or those gametes, male and female, they meet together, they form a zygote, and they have to start a new genome. We call that embryonic genome activation. So the new genes are being activated. For during those processes, one of the important thing that is happening is the epigenetic modifications.
Yeah.
And there are 3 types of epigenetic modifications mainly. One of them is DNA methylation, the second one is histone modifications, and the third one is regulation by non-coding RNAs. So what we think, or at least we hypothesize, is that because choline is a methyl donor, it could potentially be affecting the DNA methylation of the embryos. So DNA methylation, it just essentially refers to the addition of methyl groups to a DNA molecule. And when DNA is either hyper or hypomethylated, it can change the function of the DNA, but it does not change the structure of the DNA. So because, as I said, in the first 7 days of embryonic development, there is dynamic changes happening in the epigenetic program of the embryo. So it is plausible to think that if you change the availability of methyl donors during that period of development, it could probably alter the epigenetic program of the embryo, and that might have both positive or negative consequences for the calves.
That's what I was going to ask.
Yeah.
And have you seen negative?
For our feeding study, we are seeing that the calves that were derived from the dams which were fed choline, they were lighter. They were not the same as in vitro study, but they were lighter in weight at weaning as compared to the control calves. So, who knows what's happening in there? We have not done any mechanistic studies so far,
Yeah.
but we have some hypotheses that what could be going on.
Yeah.
And
Now, Marcus, I was
Yeah.
just going to say that's kind of the opposite of what you saw in your studies, right?
exactly.
Yeah.
Yes. But different, also opposite to what they saw in vitro, right?
Yes.
So really hard to,
But your studies are
Pre-
choline in late gestation,
prepare to,
Yeah.
so close up period. So,
Yeah.
but anyway.
which is a much different kind of organism.
Oh
right?
yeah, yes, exactly. It's amazing how you can modify an embryo, right? With
Right.
different compounds.
Mhm.
Talking about methyl donors, you say that choline is one of them. Have you tried others or combinations, and
Yes,
you
we
expect to
are
have the same effects?
I mean, theoretically, you should see the same effects, but these molecules, they don't just affect the DNA methylation.
Exactly.
They could be doing so many things, as I said. There are evidences that, for example, just choline can affect different cell signaling mechanisms. For example, mTOR, AMPK signaling. It can also affect the lipid metabolism. So there are several things that can happen in the embryo and which can have consequences for the calves after embryo transfer. And for the second question that you asked, we are currently working with methionine, which is another methyl donor, and we are producing embryos which are grown in different concentrations of methionine. We are looking at the effect of the concentration of methionine on the in vitro development, where we look at if it affects the percent of embryos that become blastocyst, or if it affects any of the other characteristics of the embryo. For example, if it is going to affect the lipid, if it is going to affect the impact the apoptosis, for example. And then we are also transferring those embryos to the cows to see if it has any impact on the calves. So those two experiments are ongoing.
Yeah, the
Right.
calves are born.
Right.
So we're just waiting for them to grow and get weaned. I do want to say it's kind of fun having Marcos here because he was actually the inspiration for all this research.
Oh, is that right?
Yeah, I never really had any interest in nutrients and their effects on the embryo, but I had a new grad student, Paula, who was Marcos's wife, and he was a grad student studying choline, and we were studying early embryonic development, and the whole department was just choline. And Paula, because of Marcos, was choline. So I had a new grad student come, Eliab Estrada, and I said, well, just for the hell of it, let's add choline to culture medium of embryos and see what happens. And I always thought that, we would get away from that and go back to what we were usually doing. But, it's probably been, I don't know how many years now. 6 years.
7, yeah.
So we've really gotten into choline in a big way.
Yeah.
That we never, but you're really kind of the inspiration, Marcos. So
Yeah, nice. I'm
Thanks.
glad. So I got so many questions. So are there degrees of methylation, and how can you control that, or can you control that, and what do you know at this point in time about these degrees of methylation, positive or negative?
yeah, we're doing an experiment now. Like this whole idea of fetal programming, developmental programming, is nobody has a very good understanding for how that can happen.
Yeah.
So the big explanation is at some point during embryonic development or fetal development, some key regions of the DNA get methylated or get unmethylated, and that gets inherited by the animal when it's a postnatal animal. I don't think anybody's proven that that's the mechanism. So we've looked at DNA methylation in the calves derived from choline-treated embryos, and there are changes in DNA methylation, but not just hypermethylation like you would expect if choline's a methyl donor. We actually see just as much hypomethylation. So it's not clear whether those changes in DNA methylation are the cause of the phenotype or a result of the phenotype. Maybe because the animal's growing more, some genes get methylated or unmethylated. So what we're doing now, or what we're trying to do now, we cultured some embryos with choline or without choline. We're going to measure DNA methylation in the embryo. And then we have another group of embryos from the same matings, and we're going to produce calves from those. And we'll look at DNA methylation in the calves, and we'll see how many DNA methylation sites got changed in the embryo that are still changed in the calf. See whether those are conserved or not.
Yeah.
But it could be, like Mauser said, some of these other biochemical pathways that choline is regulating, like lipid metabolism, could also be involved. So it's a big, black box really to understand how something like this can happen, right?
Exactly.
2 years after an embryo was exposed to choline, the calf's got a bigger ribeye area.
Mm.
That's pretty remarkable.
Yeah. And how much bigger would you say?
I knew you were going to ask me that when you asked us how much, and I don't remember.
Yeah. Are you seeing enough positive results from this? I think you know enough Do you see that there is a commercial implications for this down the road?
Yeah, I think so for sure. At least for the beef study that we have done, I mean, we have seen that there is negative effects, but in the embryo culture medium, we have now consistently seen positive effects, and which makes a lot of sense because in the uterus of the cows there is a lot of choline.
Yep.
You know, before as of today, there is no published data as much as to how much choline, but there are experiments where they show relative amount of choline. So they have shown that there is a lot of choline, but we have produced recently some data where we have collected undiluted uterine fluid samples and we have looked at the exact concentrations of different metabolites in the uterus. And you know, there is a lot of choline in the uterus. So that makes sense, right?
Yeah.
Because in the embryo culture medium, there is no choline.
Mhm.
So you are trying to mimic what is happening in vivo. So in the uterus, there is choline, embryo culture medium, there is no choline. So you add choline and boom, you see positive effects on the calves, right?
Mhm.
Yeah.
So that makes a lot of sense. But for in vivo, because there is already a lot of choline in the uterus, you know, it makes sense to not see any effects when we feed or, you know, see some negative effects, for example, when we feed choline during the peri-conceptional period to the cows.
So how much choline is there in the
It's
uterus?
1.8 millimolar, which is exactly what we calculated based on Marcos and, you know, his
Ah,
work and all
nice.
that.
Wow.
Yeah.
So
Interesting.
in the blood, what, it's 4 micromolar, 7 micromolar.
Yeah.
So you're talking 1,000-fold higher. Now that's in lactating cows fed
Rumen-protected methionine.
rumen-protected methionine. But
Yeah.
even in beef cows, it's hundreds of micromolar. So Vimal Selvaraj at Cornell developed this device to sample uterine fluid, and Masra improved it. And so, yeah, we have data on many, many different metabolites in the uterus. They're all higher than in the blood. Or almost, not all, but
60%.
60% are higher. All the amino acids are higher.
Mm-hmm.
One reason that we're kind of getting an interest in methionine is because of these findings of Masr's that choline's very high in the uterus, and then feeding choline is not really probably going to change those concentrations very much.
Okay.
So we think maybe methionine will be more likely to be modified by feeding than choline.
And that choline that is in the uterus is just choline or
Choline chloride, yeah.
Yeah.
yes.
This is free
Wow.
choline.
Nice.
Yeah.
Yeah, it's amazing. I mean, Paula
Yeah, Paula also
Your
showed that.
wife.
Yes, I remember. Choline was just
was one
spike.
of the
Yeah.
metabolites that was the highest.
Yes, I remember that.
So I'm kind of curious then. I mean, do we know enough today that people should maybe start putting choline in their I guess the medium, is that what you call it, for embryos?
I mean, I would say yes, because, you know, if you see an experiment effect just one time, you would be like, I don't know if it's real or not.
Yeah.
But then you repeat it again and again with 2 different beef breeds of donors, you know, that means it is real. I mean, I would definitely, if I do embryo transfers, I make IVF company, definitely I'm putting choline in there.
Yeah, yeah. Do they know about your research yet?
I think people know. Yeah,
Okay.
for sure.
All right.
You know, commercially, people don't say what culture
they're doing.
medium they use.
Yeah.
But many people put a little bit of serum in the culture medium, probably because of the growth factors that are in serum.
Yeah.
So they are adding a little bit of choline.
Yeah.
Whereas when all the experiments we do, we're using culture medium without serum. So we know exactly what's in there.
Yeah, I remember when Eliab showed
Yeah.
the first results. It was kind of nice, but the first time, right? But after 5 years, 2 trials, 3 more trials, and the same results. I think that there is something there
Yeah.
with choline. So
We do
no
need
doubt.
to do it in dairy, right? For
Yeah.
2 reasons. I think if you could get heavier calves, that would probably have a beneficial effect on the ability of those calves to make it into the milking herd. And maybe, I mean, if you look at some of the calf data, calves that grow better early in life probably produce more milk when they're cows. So that's, the practical aspect of it, I think, is very important. But there's also this question that we've never been able to answer. I don't know that this happens. Maybe the choline-treated embryo has a better placenta. The placenta produces more placental lactogen. The cow produces more milk, and that's why the calf grows more after birth. I don't know that that's true. But if it is, you wouldn't see it in a dairy animal because calf would be weaned at birth. So we've measured placental function indirectly by looking at secretion of one protein produced by the placenta. That doesn't change with choline. But I think it's still an open question. Are we programming the calf because we're programming the mammary gland? Which, of course, would be cool from a dairy perspective.
It really would, yeah.
Yep.
So is that kind of maybe, I was going to ask, what does future research look like? And you've talked a little bit about methionine, now you talked about dairy. Would that be the major thrust going forward?
We tried to do a dairy study where we were feeding just either choline, rumen-protected choline or rumen-protected methionine for the first 8 days, like Mauser described. But technically, it wasn't easy to individually feed those cows for 8 days. So we decided we would wait until Balchem develops a slow-release bolus
Well,
You're
that we
working on
could
that,
just pop
Marcos?
in. Would you agree with that, Mauser?
Yeah, I think a lot of results that you see from nutritional studies where people feed choline, they come from the system where they have Callan gates, where they individually feed the cows. And to be able to do the trial, you have to first train the cows, right?
Yeah.
And you could do that. You could afford to do that because you're working with transition cows. They are dry cows. You could just give them 5, 6, 7 days, 10 days to get acclimatized to the system, and then you start feeding choline. But since we are just focusing the period of embryonic development, the first 7 days around the time of AI, we cannot, give them 5, 6, 7 days. We have to feed them, and they have to eat it right away.
Yeah.
So that is challenging for us.
Yeah, we did try. I mean,
Yeah.
Mauser went down to a dairy in South Florida and top dress. That didn't work too well. Then he was bolusing choline orally.
Yeah.
But, that was a little stressful on the
Yeah.
cows.
for the
Yeah.
cows, yeah.
Could you do it in a robotic feeder, I'm sure? That would
you
be an
could.
option.
Yeah.
Yeah, if we could find that dairy where if people supplement some of the concentrate when the cows are milking, because you don't need to feed a lot of choline. It's
Right.
only 60 grams of choline, which is like maybe this much, right?
Yeah.
So you could make a pellet or something, and you could feed them that way. But we need to find a dairy like that and who would like to work with us.
Yeah.
And would you expect to see the same results, better results? I think that the demand of choline on that cow probably is higher. I don't know, based on meat production, other things, but who knows, right?
Yeah. I don't know. I mean, we have to do the experiment to answer the question because, as Dr. Hansen mentioned, it is also a different physiology because of the mom or the cow not feeding the calf. So that might change the results. I mean, we don't know.
Yeah.
So we have to do the study.
Yeah.
What
And-
about your sheep results?
Yeah.
Yeah, I mean, we have recently conducted an experiment with sheep. We got very much interested in sheep because it has just 5 months of gestation length. And if you want to study effects of anything on fetal programming, you could get the calves or the lambs within 5 months. So you'll see your results. You don't have to wait for 9 months, for example. So our main goal was to see and replicate the same study we did in vivo with beef cows and see if by feeding rumen-protected choline only during the peri-conceptional period, very same design, could affect the development of the lambs. So in this experiment, because for us it was easier to bolus the sheep, so we gave them 5 grams of rumen-protected choline, Reassure, which has 60% choline chloride, that product, and we fed them 1 bolus 1 day and estimated 1 day before day of breeding. They were bred naturally by the rams and until 7 days after breeding. And after that, no treatments. Then we looked at the lambs, one of the things that we saw that was very striking, that was feeding choline during the peri-conceptional period skewed the sex ratio of the lambs.
Yeah.
Really?
Right. We saw more female lambs from the dams that were fed choline. The first time we saw it, we said, this could just be a fluke, right? So we repeated the
I
experiment
said, that's
again.
not real. Monster. Come on.
He did not trust me. So I told him the lambs are there and they have everything still there on them. You know,
Yeah.
you could see if they are males or females. I didn't change anything. So
No, it's just chance.
yeah, so we did the experiment again, with more number of sheep, because the first time we had about 53 sheep. The second time we had about 60, 70 more sheep. So now we have a good N, and we saw exactly the same effects.
What is the
Wow.
ratio?
We get 60% females, about 40% males in choline, and in the controls, it's about 55, 45.
Yeah.
So
And so what's the hypothesis behind that?
you answer that. It's a tough
Well,
one.
it's interesting. I mean, unlike the cow, they have more than one lamb.
Yeah.
I forget what the average litter size was, like 1.7, 1.8.
Right.
So, but there was no effect on fertility. The fertility was the same. The litter size was the same. So it's not like choline is killing off the male fetuses.
Yeah.
So the only explanation I can think of, which I'm still struggling with, how could you get more females if you're not killing off the males? You're either preferentially increasing survival of the X-sorted sperm, which seems kind of odd, or maybe you're affecting ovulation time. There's not very good evidence, but some evidence that if, at least in cattle, if insemination occurs right at the time of ovulation, you get more males, because the Y-sperm is a little smaller, goes up the reproductive tract quicker, so you're more likely to get a male. But then those sperm die, and
Okay.
so if ovulation is delayed, it's the X-sperm that are still alive. It's only a small shift. So maybe that's true, right? Maybe the choline is affecting ovulation time relative to estrus, and so how many living X-sperm versus Y-sperm are there at the time the oocyte comes down the oviduct? Maybe that's responsible. But of course, unlike the cow studies, that ram is mating the female probably, possibly multiple times. We synchronized the sheep, so he had a lot of ewes to
Yeah.
mate. But my guess is he bred a lot of them more than once.
Right.
But it is very...
And do you have any blood metabolites from that sheep study?
Yes,
Anything that
we
changed?
did a preliminary experiment where we fed them same bolus of choline, and we collected blood sample before bolus feeding and then after every 2 hours until 10 hours. We didn't see any plasma choline going up, but TMAO,
Wow.
it went like boom. It was like fourfolds at least high in choline. So, I don't know if that is affecting the sex ratio or the embryos or the semen. I cannot tell because there is no data on that.
And we saw no effect on birth weight or weaning weight.
Yeah, no
It just
effects on
stays
birth
similar.
weights, weaning weights.
Okay.
Mhm.
Did you look at the carcasses? Any differences there?
Yes,
We-
not the carcass. We didn't slaughter them, but we looked at the carcass ultrasound,
Yeah.
right? We did ultrasound to look at the ribeye and the intramuscular fat. Only the longissimus dorsi muscles area was higher in the choline-supplemented lambs, and the lambs produced from choline-supplemented dams.
Hmm.
So that was, yes, higher.
Interesting.
Yeah.
Any other species you're going to look at? I mean, I'm wondering about monogastrics, right, and what implications that may or may not have.
Yeah, your question, is it commercially relevant to be doing these kind of things? I think so. We've never, like I'm always saying, if you look at the improvements in animal production in the 20th century and into today, it's because of genetics, but it's also because we've gotten so good at optimizing the environment of the animal. But it's always after it's born.
Yeah.
So now I think we can start to think like Marcos's data as well in late gestation. How can we modify the environment of the animal before it's born? What's tricky, it's a pure guess, right? There's no, it's hard to predict, oh, I think this molecule will have a positive effect if it's added at this time.
Yeah.
It's pretty much
Try and error.
trial and error.
Exactly.
But I think when we find molecules that do have beneficial effects, that'll have a lot of relevance to the producer.
Yeah. Marcos, we've referenced your research a couple times. Can you give us just kind of a real high-level overview of what did you do and what were some of the results that you found?
Well, the main goal of our trial was always to feed rumen-protected choline through the transition
Yeah.
and look at the performance of the cows, right?
Right.
Meat production, components, etc. But because we are working with transition cows, we start focusing on the heifers, basically, the calf that were born from moms supplemented with choline. And just why we start looking at that, because during my PhD, or Paula's, my wife PhD, we have the second baby here. And so we start looking at what minerals or vitamins a mom should start taking, and choline was just there, right?
So she was taking choline.
Oh, yes.
Yeah. There you go.
I take choline.
So everything
So
started
I'm
there.
so handsome.
And there was so many data showing good result of feeding choline to pregnant mom,
Yeah.
to the babies, that we decided to start looking at the heifer, the calf, right? Basically what we saw in 2 trials that we did, better growth rate through the weaning period, even after that, better colostrum from the moms. We tried to isolate the effect of colostrum, and again we saw the effect of the calf on growth rates in the first 60 days, less diarrheas, better immune system, if we can say that, right? So there is something going on then. Then after that, Michigan and Wisconsin follow up with 2 recent trials and similar results.
Yeah.
So we need to follow those heifers into the first lactation probably, but probably they will do better than the controls.
Yeah,
No
good.
doubt.
So I'm sure the data that you were referencing before on humans and the impact on children was the Cornell data. And then in that study, mothers that consumed choline had children that were smarter, right,
Yeah.
for the most part. And those children are now 14 years old, and they're still following up with those children right now. And that data is going to be published, but I believe that result continues on. And so my question to you is, I'm sure your children are brilliant. Is that
They
true?
are, yeah.
See, I think my mother had cigarettes and alcohol.
That's my line. I always say my mother didn't take choline.
Well, but they didn't know. I
right,
mean,
they didn't
right?
know.
People
No.
didn't know.
But talking about cigarette and alcohol, there are plenty data that choline can reverse that effect.
I know.
It's
I saw.
amazing. It's
Yeah.
amazing, the molecule of choline. I mean, it's when you start reading, it's amazing.
Yeah.
Well, I mean, there's effects on brain health. I mean, that's
Yeah,
why I
yeah.
take choline.
Well, so do I, because
Yeah.
I have an aging brain, and there's some.
too.
So, Masru, anything else that you think we ought to cover while we're all gathered here?
I think one thing that hopefully we will follow up on is, if you feed choline to the cows, how does that affect the uterine microenvironment? You know, that is a really good question that needs to be answered, because if there are effects on the embryos, for example, specifically, how are those effects being
Right.
being conducted? Let's say, for example, is choline feeding changing the concentration of metabolites of the choline in the uterus as well? You know, we don't know, because usually you just feed choline or whatever nutrient, you just take a blood sample and you say, oh, it went up, so we assume that it also went up in the uterus. But from our data that we have just collected on the uterine fluid, even if you see a normal concentration of a metabolite in the blood, it's already so high in the uterus. So how would feeding something change that concentration of that metabolite in the uterus?
Yeah.
You know, that is
Great
an important
question.
question
Yeah.
that we need to answer.
Yeah. And do you have plans to answer that, or?
If we get funds, yeah.
If you get the funds, yeah, there's a sore subject
And
right
time,
now,
right?
Yeah, right.
No,
What
right.
about ships? Are you planning to do something with transition ship? I mean,
Yeah, I would say we haven't talked about that.
okay.
But, you know, the one thing when we were waiting to have this podcast, I was just looking at the brochure of all of Balchem's rumen-protected products. And, you know, I just thought, man, we should test each one of these molecules for, I mean, minerals. Nobody knows what
Yeah.
Exactly.
minerals, what role minerals play in the early embryo, even just for fertility. And, you know, that would be fascinating to look at. And then like niacin, you know, again, plays a big role in metabolism. So I think there's lots of opportunity out there, hopefully for us,
Yeah.
but for other people interested in improving livestock performance to either use these molecules to improve fertility, affect fetal growth, and
Yeah.
affect postnatal function. So it is, the whole rumen protection story is kind of transformed nutrition of ruminants. So it's pretty exciting.
Yeah. Yeah. Great comments.
Yeah.
This has been very fascinating. I've enjoyed this. I really have. What I'd like to do as we get ready to close out here, I'd like you guys to just kind of give a couple thoughts that you'd like to impart to the audience, a couple key takeaways that you think they ought to think about as we close out here. And Marcos, I'm going to start with you if you don't mind.
Tonight's last call question is brought to you by Niasure Precision Release Niacin. Niacin is a proven vasodilator for heat stress reduction and a powerful antilipolytic agent for lowering high blood NEFA in transition cows. Protected with Balchem's proprietary encapsulation technology, you can be sure it is being delivered where and when your cows need it. Learn more at balchem.com/Niasure.
Well, for me, it's gonna be
Something
easy.
profound, please.
No, I mean, I have been reading about choline. I have some experience with choline, and I don't have doubt that choline is required. I mean, I would encourage people to try and use choline in the transition, even try after. Maybe some result from the field can feed our experience, so always we learn from them. So try and use rumen-protected choline through the transition. I think that is a good choice,
All right.
no doubt.
Yeah, good. Thank you, Marcos. Dr. Hansen.
I am trying to think of something profound. You know, federal research dollars are being threatened right now, which I think is a shame. And companies like Balchem have done such a good job promoting basic research. I mean, some of the things we're doing don't necessarily immediately relate to solving a producer problem immediately, but I think will in the long run. And, you know, I think humans are pretty ingenious and are constantly thinking of new ideas that maybe weren't obvious to people before. So I think this rumen protection story is one of those. So I think we're going to continue to see some big advancements that maybe nobody was predicting 5 or 10 years ago. So it's exciting to be involved with that.
Yeah,
Yep.
great comments. Masru?
Yeah, I think because I work in the area of fetal programming or developmental programming, I think a lot of things, not just choline or most of the nutrients that we feed, people just look at the immediate effect of that. Let's say if somebody is feeding choline in transition period, you want to look at the effects on health disorders, retained placenta, for example, displaced abomasum, milk production, and all those things. But now I guess with more and more growing evidence, we should look at the long-term effects of feeding those nutrients on the calves that are born from those moms, and even, transgenerational effects, because evidence coming from mice, it says that there are transgenerational effects that you see by, feeding different molecules or even, like, for example, different stress markers.
Mm-hmm.
So I think in cattle, we should also do those things.
Yeah. Great comments. Gentlemen, this has been fun. I appreciate
Thank
you
you.
joining us. To our audience out there, it's always great having you here. I hope you learned something. I hope you had some fun. And we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email at anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars takes place on the first Tuesday of every month, with the top research and nutrition topics that will impact your business. We also include small ruminant, monogastric, and companion animal-focused topics throughout the year. Visit balchem.com/realscience to see the upcoming topics and to register for future webinars. You can also access past webinars and search for the topics most important to you.